new species forms even when everyone still lives in the same neighborhood. That's sympatric speciation, and it doesn't rely on big rivers or canyons to separate populations physically. Instead, it uses other methods to create genetic isolation right in the same place. Polyploidy is a prime example. Picture a plant cell that accidentally ends up with too many chromosomes because of a hiccup in meiosis. Polyploidy is a condition in which a cell or organism has an extra set or sets of chromosomes. Poly means many, and ploidy means is the number of sets of chromosomes in a cell. We have two main types of polyploidy. In autopolyploidy, the extra chromosome copies come from its own species. See, auto means self. So an organism has multiple sets of chromosomes derived from its own species. For example, a plant species with two sets of chromosomes produces gametes with also two sets. Those gametes have now twice the number of chromosomes. See here? We see that in 2n. It's an error in meiosis where all the chromosomes move to one cell instead of separating. These gametes are then incompatible to mate with the normal ones produced by the plant and can only self-pollinate or mate with another gamete that had the same fate and also ended up with two copies. This effectively then cuts it off from its ancestors, sparking a new species, and poof, speciation in a single leap. The offspring of them then have 4 sets of chromosomes, and we call that a tetraploid. This is the 4 sets, the tetra, see tetraploid. And tetra means 4. Allopolyploidy happens when 2 different species hybridize and produce viable offspring. So it's when an organism has multiple sets of chromosomes derived from 2 or more different species. See, remember we had it before. Allo means other. If their gametes fuse and then double their chromosomes through subsequent generations, you get a fertile new hybrid that can't mate back with their original parent species. This is how crops like wheat, cotton, and tobacco originated. Polyploidy is mostly a plant phenomenon. Animal cells aren't as forgiving about major chromosomal mix-ups. Sympatric speciation can also unfold in other ways. Maybe a fish population in a lake starts diverging because some fish discover a new food source, say at a different depth of water, for example, and end up mostly mating amongst themselves. Over time, these two groups become more genetically distinct, even though they live in the same lake, and voila, 2 species, 1 habitat. Lake Victoria in Africa is a famous stage for such dramas. It's home to hundreds of cichlid species that arose sympatrically and live in the same geographic regions but evolved into their distinct forms. Here, see here, that's a cichlid right here. That's a fish. Another neat case, a Nicaraguan lake had 1 cichlid species that split into 2 types of fish with different shapes. Each specialized for different food sources. And you can see this. You have the thin-lipped cichlid right here, and then you have the thick-lipped cichlid right in here. The apple maggot fly is a textbook North American example. Originally, these flies only laid eggs in hawthorn fruit. But when apples arrived, some flies decided apple looked pretty good for laying eggs. And over generations, that whole jumping group stuck to apples, became genetically distinct, and now we have 2 populations, hawthorn lovers and apple lovers, living in the same area but rarely crossing paths. That's sympatric speciation in action. The bottom line is that physical barriers aren't the only way to split a population. Odd chromosome duplication, new feeding niches, or even a sudden shift to a different host plant can be enough to isolate a subgroup genetically. Nature is so creative with how life branches out. Good job, you guys. All right, let's talk a little bit more about some misconceptions of evolution.